Hello students of class 12, myself Shurmila Miss. As we all are going through a natural disaster, that's why we the teachers are not capable to take your class physically, and therefore some virtual classes are arranged. This is mainly your prescribed book for ISC, Srijan Biology for ISC Schools class 12 by Veerbala Rastogi. You the students should follow this book, and this is your scope or syllabus. Today I will go for the second chapter, sexual reproduction in flowering plants, structure of microsporangium, TS of anther, and microsporogenesis. This is today's topic, structure of microsporangium, TS of anther, and microsporogenesis. So let me start. Sexual reproduction in flowering plants. We all know that flower is the main sexual reproductive unit of a plant, and a complete flower mainly consists of 4 units: calyx, corolla, androecium, gynoecium. Calyx is the first whorl, corolla second whorl, androecium third whorl, gynoecium fourth whorl. The units of calyx, sepals; units of corolla, petals; units of androecium, stamens; units of gynoecium, carpel. And this calyx, corolla, these are non-essential whorls. Why these are called as non-essential whorls? Because these two whorls, calyx and corolla, are not taking part in reproduction. These are mainly supporting whorls, or these are protective in function. That's why these two whorls are non-essential whorls. Whereas androecium and gynoecium, these are essential whorls. Why essential whorls? Because obviously these are taking part in reproduction. That's why these are also called as reproductive whorls. This is the male reproductive unit, androecium, and gynoecium is the female reproductive unit. And the male reproductive unit, androecium, mainly the units of androecium is stamen. Stamens are also called as microsporophyll. Microsporophyle, that is stamen, unit of anther. And megasporophyll, obviously carpel, that is unit of gynoecium. Megasporophyll or carpel, that's a unit of gynoecium. It is female in nature, it is male in nature. Anther, stamen. One stamen is made up of anther and a stalk-like structure. This is filament. It is the stalk-like structure, filament, and this is anther. Anther mainly composed of 2 lobes. These 2 lobes are also known as theca. 2 lobes. Lobes are also known as theca, and these 2 lobes are connected by a tissue. This is known as connective tissue. This is connective tissue. And a carpel made up of 3 units. Basal solen part, this is ovary. Stalk-like structure, style, and the upper stigma. This is ovary, this is style, and this is stigma. And in cross section, these anther cross sectional view, anther looks like this. 2 lobes or 2 thickers of anther, and in each lobe 2 pollen sacs or pollen chambers are present. In each lobe 2 pollen sacs are present. So altogether in an anther 4 pollen sacs or pollen chambers are present. That is why it is known as tetrasporangiate. Tetrasporangiate anther. Why tetrasporangiate anther? Because 4 pollen chambers are present, and inside the pollen chambers obviously pollen grains are formed. Pollen grains are also known as microspores. Pollens also known as microspores. So inside these pollen chambers pollen grains are formed. So how the pollen grains are formed? This process is known as microsporogenesis. Microsporogenesis. Microsporogenesis is the process of the formation of microspores or pollen grains inside an anther. Is known as microsporogenesis. Process of formation of microspores or pollen grains inside the pollen chambers of anther is known as microsporogenesis. So, inside the pollen chamber, some actively dividing parenchymatous cells are present. These actively dividing parenchymatous cells are known as archisporial cells. Clear? Archisporial cells. So first, archisporial cells present within the pollen chambers of anther. These are mainly actively dividing parenchymatous cells. And the archisporial cells periclinally divide and forming 2 types of cells. Primary parietal cells, primary sporogenous cells, primary parietal cells and primary sporogenous cells. And primary parietal cells obviously gives rise to the secondary parietal cells. Secondary parietal cells. Secondary parietal cells are of 2 types: outer secondary parietal cells and inner secondary parietal cells. And from outer secondary parietal cells, endothecium layer is formed. Endothecium, endo, thecium, endothecium is formed. And from inner secondary parietal cells, middle layer and tapetum, middle layers and tapetum are formed. And from primary sporogenous cells, microspore mother cells, microspore mother cells, microspore mother cells or pollen mother cells, also known as pollen mother cells. It is in abbreviation PMC, that is pollen mother cell, or microspore mother cell, MMC. These abbreviations are important. PMC stands for pollen mother cell, MMC microspore mother cell. And obviously mother cell, so it is diploid 2n in nature. From microspore mother cell, after reduction division means meiosis, microspores or pollen grains are formed. First, the microspores are formed in tetrads. Tetrads means obviously the 4 microspores are arranged either linear or in tetrahedral form. So 4 microspores are present, that is why it is known as tetrads, formation of tetrads. So microspore tetrads, and obviously haploid in nature, because from diploid microspore mother cell, after reduction division or meiosis, the microspore tetrads are formed. 4 microspores are arranged either linear or in tetrahedral forms. And from microspore tetrads, the mature microspores or pollen grains are formed. Mature microspores or pollen grains are formed. So this is all about the formation of microspores or pollen grains, and this whole procedure is known as microsporogenesis. This is the formation of microsporogenesis. In my next video, I will discuss about the functions of endothecium, middle layer and tapetum, and obviously I will discuss the diagram of microsporogenesis, how you people will represent the diagram of microsporogenesis. In my next video I will discuss that. Okay, thank you.